This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Hi everybody, good afternoon. And welcome to our 10th week of Algorithms for Functional Genomics. So 3 weeks to go. Everybody ready for the final exam? No? Okay. Still 1 month to go. So it's still recoverable. So we have 3 topics into the end of the subject. This week we're going to talk about variant analysis. Next week we'll have the pleasure to have 2 invited lecturers talking to us about single cell analysis. And we'll finish up really strong with supervised learning. Okay? And so this week we're going to talk about variants and mutation analysis, a very important topic these days. So when we talk about mutations, what's a mutation? First thing. So Grace, what's a mutation? Yeah. I told you for the first time I mentioned this to you. It was your fault. You nominated yourself to be representative. Now you know your name. Change where? It's a change. Yeah. So basically, mutation is a change in DNA of a given organism. And basically, we do have a lot of molecular machinery to recover from mistakes while duplication. And basically, mutation is when DNA repair processes can't rescue it. And it gets fixed, and that population of cells will then have a different genome from the rest. Okay? So, and we have another concept, which is a variant. So what's the difference between a mutation and a variant? Does anyone know? So what's a variant and what's a mutation? They're both changes. But what's a variant? Not sure? Okay, so the variant is a mutation, but it's a mutation that has been fixed in a population. So typically, when we're talking about variants, we're talking about a mutation spread in a population. Okay? That's basically the difference. So you can say that a mutation with a population of frequency of more than 1% is actually a variant. Okay? Sometimes we use them as synonyms. They're not quite synonyms, but they were tried to focus on the word mutation, just to be more generic. And basically, when we're trying to understand mutations and variations, we're doing comparative analysis of genomes. So we want to understand the differences between similar genomes. And in our case, usually we want to understand differences between DNAs being studied. So we have a sample that's being studied, and we have a reference genome. We want to ask the question, what are the main differences? What are the mutations? So mutations, as this figures basically given away, it has to do with inserting diversity into genomes, which is something really important for evolution. I'm probably far bent on this side, so I'm on the T-Rex position already, so computer sciences usually are on that position. So mutations are a very important tool that nature uses to insert diversity. And they can be advantageous, they can be deleterious, they can be neutral. So for instance, a deleterious mutation could be something that increases the risk of a given disease. An advantageous mutation could be something that gives you an important characteristic, that, for instance, that increases fitness of a given organism, so the chances of it to succeed. Or they can just be neutral, and they happen by chance, but they do play an important role in evolution. So genetic variability is basic composition of the mutations and the genetic recombinations. And basically, how does then mutations occur? Mutations can occur due to external agents, for instance, exposure to mutagens, X-rays, chemical agents, and so on and so forth. So exposure to mutagens, and also due to a natural process of getting old and our DNA repair machinery not working properly. So mutations can occur naturally, and they eventually can be fixed in a population. And then we'll call it a variant. And we can broadly classify mutations into 2 types. So somatic mutations and germline mutations. What's the difference between them? We already covered this on week 1, but it's been a long time. So what's a somatic mutation?
Just one
Yeah, so that's one of the differences. So germline mutations are passed on to offspring, while somatic are not. Why is that? Why they're not passed on? They're somatic cells. So something to be passed on, it needs to be on your gametes, basically. So if you have a long time exposure to sunlight, you're likely to, or more likely to develop skin cancer, for instance. There's something that it's on a specific spot on your skin. That's not going to be necessarily transmitted to your offspring. So somatic mutations are the ones you acquire during your lifetime. It's the ones we accumulate over time. And they occur during DNA replication preceding mitosis, so somatic cells. And in that case, all descending cells from that one are affected, but not necessarily offspring. So the main thing for somatic mutations, they don't get passed on to offspring. And they are very important, for instance, in driver mutations in cancer. So driver mutations are actually mutations that will be the trigger for the cell to lose control of proliferation, for instance. And that's quite important for cancer. So somatic mutations in cancer and tumor suppressor genes, they tend to be drivers. And there is a lot of people researching it, trying to understand which are these mutations and can we use them to actually develop better methods to predict whether someone has an increased risk of developing cancer, for instance. While germline mutations, those are mutations we're born with, so they're inherited, and they occur during the DNA replication in meiosis, so in gametes, and they will affect not only the gametes, but all the descending cell lines. And therefore, it's passed on to offspring. And these are the type of mutations that are usually involved in monogenic diseases, in inherited diseases, and also in cancer predisposition as well. And one of the things about mutations is that even though we share a lot of genetic codes, anyone in this room will probably have millions of genetic variants between one another. And in fact, if you look at the average, so on average, each human has around 3 million germline variants when compared to the reference genome. So there's a lot of variation that comes up with a lot of different phenotypes and makes up for all the differences we have. And you can also divide mutation types into, depending on the size of the change, we have mutations that are characterized by larger changes, for instance, structural variants, and that these could be changing in copy number variation. So in one generation, you have 2 copies of a given gene, and the other you have 3, for instance, and you can also have deletions and inversions. While small changes, the main class of small changes are single nucleotide polymorphisms, or SNPs, is when you change only one base pair. And you can have a substitution. You substitute one base pair to the other. You can have an insertion. You can have deletions. So usually these are the main, one of the main classes of mutations because they're very small. So they're more likely to be incorporated in a population than something that is very, very drastic. So visually, what we have is something like this. So we have single nucleotide variants. This could be interpreted as DNA spelling mistakes. We have the same number of base pairs, but we change this from a C to a T. We can have a small insertion. Let's say here we have an insertion of 2 base pairs. That's extra or missing DNA, and we can have large structural variants, so large blocks of extra missing or rearranged DNA. And since these are very important, we'll try to focus on them. So single nucleotide polymorphism, just changing one base pair. So changing one base pair, even though it's a very, very small thing, that can have quite drastic biological effects. So what are the biological effects that one change can have out of 3 billion? So we have 3 billion base pairs. If we change one, we can have a biological effects. What type of biological effects, for instance? That's one thing, but that depends on basically where they occur. So you're correct. So if they occur in a region that encodes protein, so a protein coding gene, then we can have, or we may or may not have a change in the protein. But that depends. Depends on the type of the change and depends on where it occurs. If it occurs on a protein coding region, this may affect protein structure and function. Okay? But how? We can ask the question, how a mutation is affecting a protein and by which molecular mechanism? One thing is to say, okay, this codon has changed the residue. The other thing is to say, it's changing the residue, having a change in the protein structure, and this is affecting, for instance, protein stability, which is important for protein function. We can ask the question, what's the molecular mechanism of this NIP? It might have something to do with the interactions the protein does in a structural level. For instance, you have proteins that interact. You have here in blue and green 2 different protein chains that come together to perform a given function. If you have a mutation in the interaction part between them, then you can perhaps disrupt this interaction and therefore protein function. So understanding whether a mutation is going to affect protein function or not, it has to do also understanding their potential molecular mechanism. For instance, if a mutation binds to DNA, whether a mutation is going to affect the affinity for that molecule as well. So in terms of interactions, we can consider that mutations might affect other proteins or interactions with other proteins, interactions with nucleic acids, metals, and ligands, and that includes drugs as well. So a mutation can make a drug less potent or completely ineffective. And that gives rise, for instance, to drug resistance mutations. Okay? On the other hand, if we have a mutation on a no-coding region, what could happen potentially? So 2%, more or less 2% of our genetic code encodes proteins. The rest we know encodes several different things, but most of the part we don't know. So, but on non-coding regions, we know that there are a lot of regulatory elements that can be affected as well. And however, we're going to focus, especially on the next lecture, on mutations that affect protein structure because proteins are so important. So they are the macromolecules that orchestrate life in the molecular level. And they are not only extremely versatile in terms of the functions they perform, but they are also very, very important drug targets. So most of the drug targets, 80–90% of them, are actually proteins. So we want to be able to, using computational methods, ask the question, if a mutations occur here, so this is a BCL inhibitor, if a mutation occurs here, is it still going to bind? It's still going to be an effective drug. And if you think about it, we usually have 2 to 3 million variants between each other. Drugs are not developed for a person. It's developed for a population. So we have mutations between each other, meaning that we respond differently when it comes to drugs, when it comes to adverse effects. So can we ask the question, what's the most appropriate treatment for someone, instead of just treating the disease, not the patient? So understanding mutation effects in the context of the protein structure is very important, not only to understand better treatments, but also to guide and to inform drug development. If you think about it, it takes a lot of effort, a lot of time, and a lot of money to develop a drug. And that usually, well, I think the average these days is 10 to 15 years, $1 to $2 billion. And that's considering a population. There has been studying shows that, well, usually those populations are male and European, that if you consider subpopulations at admixed African, the drugs don't work as well. If you consider on a person level, that's even stronger. So we need to be able to inform drugs that work for everybody, and we need to inform drugs that are less prone to resistance. So basically, drug resistance occurs if you're trying to kill a bug, let's say with an antibiotic, and, well, the pathogen is trying to survive. And what it could happen is a variant or a mutation that happens that, well, it basically prohibits the drug molecule to bind and to kill the bug. And then you have a drug-resistant strain. So how we can use then computational tools to develop better drugs that are less prone to resistance. Even though it takes 10 to 15 years to develop a drug, usually we see resistance in 1 and 2 years. And that's very common, even with drugs that are kind of neglected and occur in very poor regions, for instance, Mycobacterium tuberculosis. So tuberculosis still kills hundreds of thousands of people a year. And it usually mostly in Africa, India, so big pharma is not very interested in investing money to develop drugs for Mycobacterium tuberculosis. In 2012, there was the first case of a variant of TB that not a single antibiotic could kill. So it was a completely resistant variant. And it took 20 years to develop a new drug for TB. So I think we can do better than that. And bioinformatics and computational biology have a extremely important role in that, into trying to develop better drugs. And I think I digressed a little bit, sorry. And just because it's something that is quite interesting. And so we're talking about SNPs in protein coding regions. And we know that our genetic code is what? What's our genetic code? Our genetic code is basically how codons encode residues, amino acid residues, and a few other things, right? So each 3 base pairs encodes potentially 64 different words. However, we have 20 different residues. So our genetic code is redundant or degenerate, okay? And because of that, we have different SNP types. So we're talking about single variations, so single variations in a codon. And depending on the type of a variation, will have a different effect. So what types of changes we can have for a given SNP? So I changed one nucleotide on a given codon. What's going to happen to the protein? What are the possibilities? First possibility, what's going to happen? Nothing. So synonymous. So we have one type of SNP which is called synonymous SNP. For instance, if we have a lysine, where's lysine? Lysine here, okay. So lysine has 2 codons, AAA, AAG. Let's say I have an AAA and it's changed to AAG, it's still going to encode lysine. So our protein, our gene product, our protein will be the same. So does it say that nothing going to happen to the organism? The protein is the same. So if it's an enzyme, it's still going to encode a functional enzyme. Is that going to be the same thing as the wild-type variants? Not necessarily. So there's something called codon usage. So that could be affected. So the protein levels could be changed, even though you might have enough functional protein. The alternative to synonymous is, of course, non-synonymous. So something is changing. So I'm changing from one residue to something else. And non-synonymous SNPs can be further divided into 2 classes. Nonsense is when I change a residue to a stop codon, which leads to protein truncation, which most of the time the protein is non-function. So let's say you have a protein with 500 amino acids, and on position 300 you have a nonsense mutation, you lose all of the rest of the protein. And that's related with a lot of inherited conditions. One famous one is related to cystic fibrosis, which is one mutation, one base pair change out of 3 billion, which is on a glycine on position 542 that is substituted by a stop codon, and that causes cystic fibrosis. And we have a different class of non-synonymous SNPs, which are missense mutations, which are quite interesting because they are could have very subtle effects. So on missense mutations, I'm changing one amino acid residue to another. Let's say if I change AAG to AGG, so I'm changing from lysine to arginine. That's a missense mutation. And missense mutations can be divided into conservative and non-conservative. And they're more likely to be incorporated in the population given that they might have subtle effects. Okay? However, their effects are very difficult to predict. A lot of diseases and inherited conditions that actually occur due to mutations that can have very small effects on the protein, but that's enough to trigger a disease or a pathogenic mechanism. So basically, just to clarify, well, if we consider we have our codon here, AAG, that encodes for lysine, a silent mutation or synonymous mutations will be changing a codon without changing what's in protein level. A nonsense one would be substituting that codon for a stop one. And a missense mutation could be either conservative or non-conservative. What's a conservative mutation? Similar what? Similar amino acid in which sense? Shape? What else? Yep, so physical chemical characteristics in general, for instance. This is conservative, right? So you're changing from lysine to arginine. They're both positively charged. If you think if it's a very important residue that needs to be charged, for instance, if it's doing an interaction with a negatively charged one, that can be considered a conservative mutation, while a non-conservative is changing the characteristic, for instance, in this case, a threonine. Then you basically lose the charge, okay? And these are quite important ones. And for instance, conservative and non-conservative missense mutations are reflected in substitution tables, for instance. What do you expect value to be in a Blossom matrix for a conservative mutation? And for a non-conservative mutation? That's a really good exam question. I should have thought about it. Maybe I did, maybe I didn't. You'll know in a month. So why do we need to study mutations? Why do we need to study this? So mutations can give rise to very different phenotypes. We already mentioned about disease and disease predisposition, but also mutations are related with cancer predisposition, inherited diseases, and drug resistance as well. So very different mechanisms and very different things, but on a molecular level, you can consider them in the same way. So you can ask the question, what's the molecular mechanism of this mutation, regardless of whether it plays a role in drug resistance or in cancer predisposition? And what we need to do, what scientists need to do, is to understand and develop tools to predict mutation effects. Because understanding the mutation effects is the first step towards personalized treatment, towards predicting disease risk, towards patient management, and so on and so forth, and pathogenesis mechanisms. So this could help. If we can understand how mutations occur, if we can distinguish between pathogenic and neutral mutations, then we can better inform patient management, for instance. If we can predict mutations that cause the drug resistance even before they occur, we can inform the drug development pipeline to say, oops, be aware that you're developing a drug that it's likely to cause, to arise resistance. And you can think about predicting disease risk. For instance, how nice would it be if we sequence your genome and be able to say, you have a 95% chance of developing this disease or that disease. So perhaps you need to be extra careful. So for males, for instance, you have a really, really high risk of developing prostate cancer. You need to take care of that. And the same thing for women. And there have been a lot of development in that to try to understand risk looking to our genotypes. And that allows for a better patient management. But also in terms of patient management, is actually something to do with individualizing treatment and personalized treatment, so treating the patient, not the disease. So considering your genetic background in order to choose the appropriate treatment for you. So I mentioned tuberculosis, and that actually is happening now in Victoria. So usually for tuberculosis, what happens is a bug that takes a long time to grow. So in order for you to experimentally assess whether it's resistant or not, in order to choose which drug to use, that takes months. So what they usually do is, let's start with the treatment. And that's a nasty treatment that takes 6 months. And usually if you don't have access to actually phenotyping it, after 6 months, sorry, there's a lot of side effects and it didn't work. Now you're going to put on a second-line drug, multi-drug resistant. You go from 6 months to a year and a half or even more. So what if instead of that you can have a computational tool that quickly can say, you sequence the patients. For instance, they've been doing this with sputum. So they sequence the sputum, they identify the mutations in TB and say quickly with a computational program, this is likely to be resistant. Then you select the actual treatment. And that's being implemented currently in Victoria. So in a way to use computational tools that look at the mutations and can quickly predict, even if they never occurred, using, for instance, machine learning, the most appropriate patient treatment. And that has the effect of basically you're saving money in terms of the treatment, and you're actually improving patient quality of life. Okay? So the main goals for this lecture and the next one is basically we're going to talk about how do we get variants, so how do we detect variants in mutations experimentally, and that has to do, for instance, using SNP chips or SNP arrays, and, for instance, using sequencing, next-generation sequencing. And if we do use next-generation sequencing, given our sequence sample, how do we identify variants? Looking at the reference genome, and that has to do with variant calling. And this is today. And for Friday, we want to go into the actual methods used and have been developed over the last few years to ask the question, how do we predict effects of missense mutations in proteins? Okay? And we're going to talk about computational approaches, both of them, those that look just at the sequence information, and those that use at the structure information to quantitatively and qualitatively predict effects of mutations. Qualitatively will be something like, I've observed a new mutation, is this pathogenic or not? Is this likely to cause resistance or not? While, for instance, is this given mutation a pathogenic one or a benign one? And quantitatively is how much of this mutation is actually affecting protein stability or protein interaction. So how do we go about using experimental data to build new tools to understand and interpret mutations, and a few of computational resources in terms of mutations databases that can be leveraged to build new tools, or also to look up for known variants that have been experimentally characterized as pathogenic. For today, we're going to focus on how do we detect variants from SNP chips and next generation sequencing. So variant detection using SNP chips. So what we can do with SNP chips is identify or detect known SNPs, the SNPs that we've seen before, or SNP that for which we tailor probes to do it. So this is also called a technique called SNP arrays or SNP microarrays. And the main idea is that the microarray will be a set of known DNA sequences or probes that are fixed to a surface. And these probes are single-stranded oligonucleotides, and they are short and manufactured DNA molecules. So they're tailored to identify specific SNPs, let's say, in specific genes. And variant detection with this will be the following. So in order to use it, we'll basically fragment the target DNA we have, and we basically label these DNA fragments with fluorescent dyes by attaching fluorescent dyes to the end of it, and we'll wash our target DNA over this array, which has a lot of probes there. And we'll use basically an imaging instrument to check whether we have any complementarity to the probes. And given that the complementarity will cause the DNA to bind and match the probes. So we can actually identify whether a given sample has or has not a given part of DNA. And for SNP chips in particular, what we could do is actually have probes with our reference sequence and with our variant sequence. So what we expect is that sample DNA will bind exact complementary with probes more strongly with mismatched ones. So this complementary bind with the reference will be stronger than the variant, so we'll be able to identify, and vice versa, so we'll be able to identify whether we are seeing a reference, whether we are seeing a variant, or whether we're seeing both of them. So in that way, we can detect which probe gives the stronger signal, one of them, the other, or both of them. However, this has an important limitation, which is we can only detect what we know, so what we have probes for. So this is a very very specific approach. For instance, if you were looking for a specific type of mutation on a specific protein, for instance, people have been doing that for BRCA1 mutations, which are important for breast cancer. So you just see whether you have those sets of mutations or not. However, you miss any novel mutations. This is just to look up, do you have this mutation or not, rather than discovering what type of mutation, or other mutations you have. On the other hand, if we do variant detection using next generation data, what we want as an input would be basically our reference genome and a set of reads aligned to that reference genome. How do we align reads to a reference genome? What do we do? Do we do global alignment? Interesting question too. So how do we align reads to a given reference genome? Okay, so let's say we sequence either an exome or the whole genome. We have a reference and we align our reads to our reference genome. And as an output, what we would like is a set of variant calls and a confidence information for each of those variants that we called. And unlike SNP chips, DNA sequence can actually detect not only known but also novel variants. And this process of identifying variants from sequence data is called variant calling. So let's say we have here our reference genome down the bottom, and we have all our aligned reads, and we notice there's something different here. So in this position in the reference, we have an A, but we have a lot of evidence that suggests that on our sample, we are actually seeing a G. So this is basically the variant calling format, where we're going to have the genomic location, the chromosome and the position of that particular alignment, the reference, and the variant. And in that case, we have a reference, which is A, alternative, we are seeing that's a G, and a confidence value, which is the variant score. And we also can have insertion, so we had a G and you went to GT, or a deletion from CT to C, for instance. And we want to select only those that are high quality. So we can only select variants and call it a variant if we have enough evidence. So basically, this being a high quality call, this is evidence-based. So we have enough depth and enough evidence from our sequencing to say, well, we're actually seeing a variant. Like our invited lecturer, Simon Gladman, liked to say, in science, we're not allowed to make stuff up. So we always need to use evidence. And our evidence here is depth. Well,
is the confidence only based on coverage, or
a few more things. Yeah. So I'm going to talk about I'm not going to talk about any of the specific algorithms, but the general approach, which is probabilistic. So it has to do with the depth, but it has to do with a few other things as well. Okay? So the difference between this one is that this is a low coverage, low depth, actually. So we don't have enough evidence to say, well, this is potentially a variant. It could be just a sequencing error. And the main difficulty for variant calling actually is because NGS inherently has errors. So we have errors in our data. So variant detection becomes really difficult because of the errors. So what's the difference then between this, that it's an error, and this, that it's an actual true variant? One of the things you can use, of course, is depth. So it's how much evidence we have. But we can't do anything about read errors. So they're experimental errors. But we can do other things. For instance, we can have alignment errors. For that, we can try to improve our alignments and solve that issue. But we can also consider that read errors in EGS are pretty rare these days, and every day gets better and better. However, so are variants. So variants are very rare. So if I mentioned that we have 3 million variants, 3 billion base pairs, we have 1 germline variation per 1,000 base pairs. So it's relatively rare as well. And that's for germline. For somatic mutations, they're even rarer. So even though they're very important for cancer, we have usually 1 somatic variation per million base pair. So what's the difference between this and the actual error rate from a next generation sequencer? So an algorithm that will do variant calling, that needs to identify a trade-off between 2 things: sensitivity and specificity. What's sensitivity and what's the specificity? Anyone knows what sensitivity is?
How many percentage of the true variants have you detected?
Yeah. And what about specificity?
Among these detected variants, which is the true variant, the real variant?
So based on everything I said was positive, how many are actually positive? It's almost that. That's what you explained is actually precision. So basically, sensitivity and specificity, they are performance metrics. So you don't need to worry too much about this for now, because we're going to cover this on supervised learning, on metrics to actually evaluate whether you, let's say you build a new classifier, whether it's how good it is, and we'll cover that on week 12. So sensitivity and specificity are performance metrics. A different name for sensitivity is actually recall, And it's basically asking the question, how many real variants do we succeed in finding? Which is basically we want to reduce as much as we can the false negatives. So sensitivity is basically the true positive divided by true positive plus false negative. So how do I make a classifier or a variant caller that is 100% sensitive? Very simple and stupid way. Yeah. If I say everything is a variant, I have 0% false negatives. I'm basically saying, oh, I didn't miss anyone. But that's not very specific. So we need to actually find the sweet spots between the two. So specificity will be how many of the false variants do we actually correctly reject. And it has to do, of course, then minimizing false positives. And we need to find a trade-off between the two. And graphically, it would look something like this. So if you look about sensitivity, it could be, for instance, how many sick people are correctly identified of having the condition. And specificity would be how many healthy people are identified as actually not having the condition. Okay? And we need to find a sweet spot between the two. So variant detection, considering next generation data, actually starts then with the alignments. And for variant detection, we almost always will have a reference sequence available. So this process is actually called resequencing. So we already have a sequence, we do the resequence, and the first thing we do is aligning the reads we have to the reference genome that we use a specific type of alignment that I'm not going to say which one it is. If you don't have a reference, however, you can always go back to de novo assembly. Right? So let's say we're going to align some reads, and we have 7 short reads of 4 base pairs each. And these are our 7 reads. Okay? First thing we do, we have our reference genome. Let's say a very small one, 31 base pairs. And it looks like this. And we start aligning them. And, yeah. And we end up with something like this. So our reference is saying that this position is a G. Our data is suggesting that it's an A on the R sample. How do we call this a variant? How do we call this a sequence error? That's the main thing variant calling algorithm will do. And variant calling approaches usually use either heuristics and or thresholds to choose variant sites. And this has to do, for instance, the number of reads supporting an alternative LL, the number of reads supporting the reference, the base qualities, strand bias, considering we're reading from both strands of DNA. But in many cases, what happens is most of the algorithms for variant calling usually actually use a probabilistic variant calling algorithm that's known as genotyping. So first of all, what's a genotype? What's a genotype? What's a phenotype? And what's a genotype? Anyone? So basically, genotype, it's our genetic content. It's our identity. And in genomics, a genotype can also be referred to an inherent allele or variant for a specific form of a gene. For instance, an individual that carries or inherits a variant from a disease-associated gene, we can call that as a genotype. Okay? And in variant calling algorithms, genotyping refers to algorithmic estimation of genotype given sequencing data. So looking at our reads and our alignments, what genotype we have. So what specific trait? While a phenotype is basically the observable characteristics or traits we have in an individual, which is a combination of genotype and environmental factors. So it's how genetics interact with the environmental factors. So humans are diploid. So we do have 2 copies of each chromosome coming from each of our parents. And when it comes to genotyping, we can have, for a given variant, 3 possibilities. We can have 0, 1, or 2 copies of a given variant. And these will make up 3 different genotypes. So let's call A our reference allele, B the alternative. So we have 3 possible genotypes, which would be AA, BB, and AB. Okay? and B is the homozygous variant. So we have 2 copies of the variant. AB is a heterozygous variant. We only have 1 copy of it. And AA is a homozygous reference with no variants. So we have 3 different genotypes for a diploid organism. While for an haploid sequence, what we have is, let's say, genotype A and B. We either have or don't have the variants. One we can call wild type, the other we can call variant. If we have 3, then we have 4 possibilities. We can have 0, 1, 2, and 3 copies, okay? And 4 different genotypes. Let's consider we're dealing with diploid organisms for now. And if you recall from sequencing, what we do in sequences is we make a lot of copies of DNA, we break them down, and we read the bits and pieces and put them together again with mapping, okay? But we don't manage to read them all, so they are sampled randomly. So what we expect to see is that the read count matches the potential genotypes approximately. So if you consider this, that we have 3 potential genotypes. So this is the homozygous for the reference, this is the homozygous for the alternative, and this is the percentage of alternative alleles. So we expect if we don't have any variants, that we don't see any alternatives. If we have a heterozygous variation, then we expect that approximately half of the cases will have AB. And if we have a homozygous for the variant, then 100% of the time we're very close to 100. So we expect that the read counts would match those things. And then based on, for instance, on the read counts, we can try to guess which is the corresponding genotype. That's the same thing for triploids. So it could be 1/3, 2/3, or 3/3 of it. Okay? So genotyping or genotype calling is basically choosing a genotype given the data you have, and one of the main strategies is finding the probability of each genotype given the observed data. So most of the methods try to find a probability. And in this case, a conditional probability looks like this. So what's the probability of having a given genotype, given that we have our observed data that comes from our sequencing, and we calculate this for all possible genotypes. If we have a diploid organism, let's say 3 possible genotypes, and we take the most likely one out of them. And let's consider this example here. So we have our reference genome, and we have our sequences, and we see a difference here in this position. So at this site, we have a deep read depth of 18, and our reference allele is C. Our alternative one, let's say, is T. And what we observe here is 11 Cs and 7 Ts. Okay, that's our evidence. So what would you guess in that case? Do we have a homozygous, alternative homozygous reference, or a heterozygous, just based on this? So one could guess that even though this is not a perfect 50-50 situation, it would have been 9 and 9, you can guess that the probability of the genotype CT is higher, given the data you have, is higher than both of the homozygous alternatives. Okay? And that's basically what's done on variant calling using Bayesian inference, and that's called Bayesian genotyping. So what we want to calculate is the probability of a phenotype, given the data we have. But first we calculate the opposite, so the likelihood, the probability of the data given we consider a specific phenotype, and then we use Bayes' theorem to calculate this probability. So just with a simple example to explain how it works, let's say you have developed a test for breast cancer, okay? That's going to basically say whether the person has or hasn't breast cancer. So your test is 98% sensitive. So 2% of the disease cases are missed, only 2%, okay? And it's 99% accurate, so 1% of the results are incorrect. And the question we want to ask is, if a patient gets a positive test, what's the chance that they have cancer? Okay, is that clear? So I'm asking the probability of positive given cancer. Okay, and for that, using the Bayes theorem, what we need to calculate is first the probability of positive, we're asking the question here, probability of cancer given a positive is the opposite of what I say. So we basically need the positive of a test result, or a probability of a positive test result given the person has cancer, and that we know is 98%, which is the sensitivity. The probability of someone getting cancer, let's consider this, that 1 in 1,000 women will eventually have breast cancer, so it's 0.001. So this is a statistic we actually need to have access to. And the same thing, in the same way for the positive test results. So in week 12, we're going to discuss something called a confusion matrix. So these are the type of probability we can extract from it. For now, let's consider that the positive probability of giving a positive result is 0.011. And this is basically composed of true positives and false positives, given the accuracy of your test and your false positive rates. And all we need to do is, with this three information, actually calculating the probability of someone actually having cancer given that they were given a positive test result. And we'll basically need to do this math here. And this will give us basically a 9%, which is surprisingly low. So only 9% chance of your positive test result is true. Why is that? So these are made up numbers, but why is that? Why so low? Since you have such a high specificity and accuracy. And that has to do with the fact that cancer is rarer than your false positive. While you have a false positive rate about 1%, cancer incidence is 0.1%. And that has the effect that you're missing a lot of things. So prior knowledge that cancer is rare is important in that case. So going back then to our calculating probabilities of different genotypes, what we need to do is calculate three things then. We need to calculate, well, we want to ask the question, what's the probability of genotype given our sequence data? We need to calculate the probability of the data given for a specific genotype, which is the probability of seeing a set of reads for a given genotype. PG, which is the prior probability of the genotype. So our prior expectations of seeing that specific genotype, either homozygous or heterozygous, and then the probability of seeing a particular set of reads. And with that, we can calculate this for each of the genotypes, choose the most likely one. So let's start with PD. So the probability of actually seeing a given set of reads. So for a given data set, PD is usually just a constant. So it's the same regardless of the genotype. So instead of actually calculating the probability of seeing a set of reads, we could just normalize it at the end of the calculation. So since we only have 3 possible genotypes in this case, we can say that the probability of genotype 1 given a set of sequences plus the other genotype plus the other one, this needs to be 1 because we only have 3 possibilities. Okay? So instead of calculating PD directly, we only need to calculate PD given G and PG. Basically because we can express this like this. Okay, so we don't need actually to calculate this directly because it's going to be constant. If it's going to be constant, we can just normalize it after the calculation. What about the other bit, which is calculating a probability of seeing reads for a given phenotype? And what you would expect on our example is that we have a read depth of 18. We've seen 11 Cs and 7 Ts, and we want to ask the question, what's the probability of seeing this data for each of the 3 possible genotypes? And what we could do is, basically for homozygous genotypes, what we expect is that if the data is all A, we're only seeing A. If the data is all B, we're only seeing B, except when we have reading errors. So if you, for a given genotype A, all we want to see is A. If we don't see it, it's probably a sequencing error. While if we have a heterozygous genotype, we're basically choosing, given the process of how next generation sequencing works, randomly an instance from A and B, and this will end up generating is a binomial distribution with half and half probability from one and other variants. And then the last part, which is the prior probability of a given genotype. If genomes were random, we could assume that the probability of this specific genotype is 25%, this specific one is 25, and a heterozygous is 0.5. Is that clear? That is if genomes were random. But we know that that's not the case. This is assuming that variants are as likely to occur as non-variants. All right? And we actually expect, when we sequence someone's genome, to see most of things conserved. So one thing is that the genome is not random, and most our genomes are very similar. So we usually expect to see the reference genome sequence. And for instance, considering each human has one SNP per kilobase, one variation every 1,000, we have 1 in 1,000 chance of a variance in a given site. So it's definitely not random. And if each chromosome's allele is chosen randomly from the population, then if we just have one, we have 1 in 1,000. To have two is actually 1 in a million. So in other words, variants are rare enough that we need a lot of read evidence to confidently say that what we're seeing is real. And if we have a sequencing platform that for which the sequencing error is very high compared to the true rate of variance, we basically will not have enough data or enough depth to say it. And in that case, we can't really tell the difference between false positives and true variants. So having good quality data is extremely important to call variants. Otherwise, you won't have enough evidence. You won't be able to tell the difference. Okay? And that's the basic principle of variant calling. And I think that's it for today. No, we have something else. So we can do more things. Almost there. So we can do more things to improve our chance of getting variants. One of the things is actually extra steps to decrease the false positives. One thing we can do is try to improve our alignments. And before alignments, trying to improve our reads, so removing reads that for the region that the quality is not very good. That's one thing. The other thing is actually improving our alignment, making sure we don't have any mistakes there. And we can, after filtering, using filters to remove variants that are a bit strange. And there has been a lot of development in that area, and there are a lot of tools available to do variant calling. For instance, the GATK from the Broad Institute that will allow you not only to do the probabilistic variant calling, but all the pre-processing processing steps as well. And now that's it. Thank you very much.